Hello and welcome to the first episode of the new Innova Biosciences webinar series. This webinar is in association with Chromosite, the Flow Cytometry web portal. Today, Professor Graham Pockley from Chromosite and Dr. Andy Lane from Innova Biosciences will introduce you to various aspects of flow cytometry and all you ever need to know about preparing fluorescent conjugated antibodies. My name is Claudina, and I would like to take a moment to explain the structure of this talk. Our first speaker, Professor Pockley, will give you an introduction to flow cytometry, the history behind it, the necessary equipment, and the applications of the technique. Our second speaker, Dr. Lane, will discuss antibody labeling technology in relation to flow cytometry. In case you're interested in drilling deeper into certain aspects of this technique, we have allocated time at the end for a question and answer session. Please feel free to type in questions into the questions box in your control panel throughout the webinar. In the Q&A session, we will answer as many as we can, but if we run out of time, don't worry because we will respond to the remaining questions by email. Well, that's the basics laid out. Now, please welcome Professor Graham Pockley.
Thanks, Claudina, and hello everybody wherever you are. What I would like to do in the first part of this webinar is really just introduce certain basic aspects of flow cytometry and we'll build on the complexities of the technique as future webinars are released and we move forward with providing you with the information that you need to design and produce excellent experiments. Okay. So it's always good to start with a definition, and really I just pulled this off Wikipedia. So flow cytometry is essentially a technique for counting, examining and sorting microscopic particles suspended in a stream of fluid. It's important to note that really flow cytometry is particle analysis, so you don't necessarily need to use eukaryotic cells. You can analyze bacteria and a number of other parameters within the technique. But today we'll really be focusing on cellular analysis. What flow cytometry allows you to do is analyze large numbers of cells very quickly indeed, which sets itself aside from the microscopical based techniques. It also allows you to measure multiple parameters on a single cell, which again has an advantage over the more commonly used flow microscopic fluorescent techniques. Okay. So I thought it would be just worth highlighting some applications of flow cytometry, but this really isn't the complete list. For example, flow cytometry can be used to analyze DNA in cell cycle analysis, cell proliferation, multicolor phenotyping, as well as characterizing how a cell looks, so to speak. You can do a lot of functional assays using flow cytometry. And in my own lab, we do a lot of monocyte, oxidative burst, and neutrophil functional analysis, as well as natural killer cell cytotoxicity assays. And these assays can be developed for other cytotoxic-based studies. So this is only a very short list, but as I say, you can use flow cytometry for many, many applications. I think it's always good to look back in history and sort of identify, who were the main players at the beginning of any technique, really. So the first fluorescence-based flow cytometry device was actually developed by Wolfgang Goede in 1968. And those instruments were first commercialized by a company called Partec in Göttingen. Those machines and those instruments are still available. The original name of flow cytometry technology was actually Impulse Cytophotometrie in German, and this was changed to flow cytometry at the conference of the American Engineering Foundation in Florida in the late 1970s. The ability to measure multiple parameters such as volume, light, scatter, and fluorescence, which we'll come back to later on, using a single instrument was developed by Paul Mullaney, and the capacity to measure side scatter was developed by Gary Saltzman. Mike Fuller, working in the Los Alamos National Laboratories, developed the first cell sorter in 1965, and Leonard Herzenberg first coined the term fluorescence activated cell sorter, or FACS, in the mid-1970s. I'll come back to that later in the session. So those of you that have already started flow cytometry probably won't really recognize any of these instruments. These are the first instruments which were developed in the late 1960s and early to late 1970s. And actually what you'll first notice, apart from the size of these instruments, is the fact that they typically required a specialist operator and a dedicated facility. And they also could really only sort of look at 2 or 3 parameters, 3 colors in the maximum. Clearly things have moved on since those days, and we're now blessed with a large profile of instruments provided by a number of different suppliers, such as we have listed here. And actually, the development of the machines, although they've gone down the route of making these instruments extremely accessible to the user, the ease of use has to be treated with caution, because it doesn't replace the need for understanding the principles behind flow cytometry and the need for the appropriate control experiments and control samples within each of those experiments. And really, as we develop this webinar series, as Innova Biosciences develops this webinar series, we want to build on developing a better understanding of how these instruments work and what kind of information you need to know to make sure that the data you're generating is valid and appropriate for the use of the instruments. But before I move on, I think it's just worth highlighting that you don't necessarily have to be in a laboratory to analyze cells using flow cytometry, and I just thought it'd be worth highlighting these 2 instruments, the CytoSub and the CytoBoy. The CytoSub is a submersible flow cytometry which is produced by a company called CytoBoy, and CytoBoy sits on the cell surface, and these instruments are primarily used for monitoring plankton flow in ocean currents, and so it's a very widely usable technique. And it's also the case that these instruments can be used in the field, so to speak. And here we have an instrument which is produced by Partech, and this is really a POC, a point of care instrument, which allows HIV and AIDS immune status to be monitored in remote and rural areas by providing a basic CD4 positive T cell count. And this particular instrument is battery operated, and so this allows remote analysis to be undertaken. Okay, so if we now move on to the basic sort of components of a flow cytometer, these are highlighted here. Essentially you need a light source, which is typically these days is a laser light source, which I'll come back to, but any light source, other light sources are available. The other key element of a flow cytometry system is a flow chamber, and that's the chamber in which the light source hits the cells in which you're interested. And again, we'll go back into that in more detail in a few minutes. One needs an optical system, which allows the collection of the optical signals. We also need the light detectors, which allow those optical signals to be converted into electronic signals, which can then be collected and analyzed by the computer and the associated software. So if we look at the flow cell, for instance, a very sort of simplified version, going back to the basic principles, what a flow cytometer can do is it can analyze cells which are in a suspension. One of the key elements of a flow cytometer is focusing the mixed cell population into single cells, which can then be interrogated by the laser and the other components of the instrument. And that is typically called hydrodynamic focusing, and so the systems are there in place to convert a cell suspension into a stream of single cells which can be analyzed independently. Those can be hydrodynamic, so that could be the fluid system which generates that, but some instruments these days can now focus the cells or produce a single cell stream using acoustic techniques. So if we now look at a very simple diagram of a flow cytometer, this is just a single laser instrument. Of course, these days flow cytometers can contain multiple lasers, up to 4, 5, 6 lasers, and some typical types of lasers are listed just under here, so we can get lots of different wavelengths. The principle is the same, is that the laser light is shone through the flow chamber, which is where the cells are. Some of the light goes straight through the flow chamber and is picked up by a photodiode, and that gives a parameter called forward scatter, and I'll go back into that in a few seconds. But what forward scatter does is it's an indicator as to the cell size. So the light which has gone into the flow chamber is then directed through a series of filters and particular mirrors, and we'll go back into that later on. And the signals which emanate from that system are collected by photomultiplier tubes, and that will give you the fluorescent signals which are required for your particular experiment. So for flow cytometry, the key parameters measured are scatter and fluorescence. So in the first instance, if we look at the analysis of lysed whole blood, so these are real data which was generated by actually one of my PhD students whilst he was in Sheffield. And he was interested in examining the effect of opioids, so analgesic pain-killing drugs, on immune function. And part of his work was to analyze the effect of opioids on the phenotype, so cell surface expression, and also function of neutrophils, monocytes, and lymphocytes. If you lyse all the red blood cells and put that lysed sample through your flow cytometer, and then look at the side scatter versus the forward scatter, so this is the forward scatter and side scatter, then in this instance you can get 3 very clearly defined populations of cells, which in the whole blood are the neutrophils, the monocytes and natural killer cells, and the lymphocytes. And these are defined on the basis of cell size and cell granularity. So neutrophils are pretty granular cells, and so they are higher up on this y-axis. And the presence of these different cell populations allows one to put a region or to define that region using the software, and then you can go and analyze the function or the phenotype of cells which rest within that particular defined region. So again, I think it's useful to define fluorescence, and it's the emission of light by a substance that has absorbed light or other electromagnetic radiation. Although today we're focusing on basically antibody-related techniques, there are a number of different fluorescent molecules which allow you to interrogate different aspects of cellular function. But it's worth just highlighting what is fluorescence in a very sort of stylized way. So fluorophores, or fluorescent molecules, can be excited at particular wavelengths, and that leads to the elevation of electrons into different levels. This is an unstable situation, and the electron decays down through the levels, leading to a loss of energy. And this loss of energy leads to the emission of light. And so here, if we look at the wavelength versus relative intensity, we can see there's an absorption wavelength here, and there's also wavelength of light at a particular wavelength, which is given off as a consequence of this loss of energy in this decay. And the term that you should really sort of remember is what's called the Stokes shift, which is basically the difference between the absorption and the emission spectrum. And it's always good if you're trying to determine fluorescence to have the Stokes shift as large as possible. So what options are open to you when you start to do experiments? Well, this is just a sort of short list of the currently available fluorescent molecules, and Andy will be going back to this later on. These are the ones which are available through Innova Biosciences. But you can see that there are a number of different fluorescent molecules, all of which are excited at different wavelengths, and so these would be excited in the 405, these are excited in the green, etc., etc. And the peak emission is indicated here. So it means that if you have a cell which is labeled with a fluorescent, say dye-light 488, and you excite that cell at 488 nanometers, and you can then detect the emission at 518, and that will define the labeling of that cell with that particular fluorochrome. Okay, so staying with antibody binding, which is what we're really focusing on today, it's important to note that there are different ways in which antibodies can bind to cells. The first is really antigen specific, in which case the antibody binding site actually binds with the epitope of interest, which is here. You can also get specific binding, but it's not antigen non-specific, and that could be where the FC portion of the antibody, which is this section here, binds to FC receptors on the cell surface. And this is something you need to be particularly careful with if you're analyzing immune cells, because activated immune cells can upregulate the FC receptors, and so it is also possible that the antibody binds in this direction. Unfortunately, we don't have time today to go into all of the specifics of this binding and how you need to make sure that the appropriate controls are in the system, but that will be a subject of one of our future webinars. It's also important to note that binding could be non-specific, i.e. the antibody just non-specifically adheres to the cell surface, in which case that's typically of low affinity and it's not saturable, which means you can just keep on adding antibody and you'll keep on increasing the signal that you're getting. Okay, so if you look in the literature, you'll probably see basically one of two, or a combination of two types of depictions of the way that the fluorescence is presented. We'll come back to this in more detail in a few seconds, but basically you can present data in the form of a dot plot, which is this here, where it's 2 dimensions. So in the y-axis here, these are cells which are labeled, stained with an antibody which is labeled with Alexa Fluor 700. And on the x-axis, we have the staining profile for cells which are labeled with FITC. In this particular experiment, we used an antibody to CD8 labeled with Alexa Fluor 700, and we used a commercial antibody mix which picks out CD16 and CD56 positive NK cells using FITC. And you can see quite easily that it's possible to identify defined populations on the basis of this profile. And for example, you could put a region around this and then analyze those cells, the phenotype of those cells in more detail through a various hierarchical system. It's also possible to analyze cells using a fluorescent histogram, and these are data which reflect the expression of a molecule called CD69, which is an early activation antigen on CD8 cells which have been unstimulated in blue, or which have been stimulated using polyclonal stimulating beads in red. And so you can see that stimulated cells express, there's a population of cells which express CD69, which confirms their activation status. Okay, so when it comes to sort of a very basic way of analyzing the data, here we have just a sort of stepwise way of depicting this. So again, we're looking at a sample of lyzed whole blood, so we have the lymphocytes, the monocytes and the neutrophils here. We're particularly interested in the lymphocytes, so we've put a region around the lymphocytes and we look at the fluorescence characteristics of this. the first step is to have a look at the cells which haven't been stained at all, so the autofluorescence controlled as it's called, because that's a way of actually setting the negative on your instrument. It's not what you also need to include in some instances is an isotype control which identifies the non-specific binding, and an isotype control is really an immunoglobulin which has been demonstrated not to bind to your cells but is labelled with the same fluorescent molecule, and so it gives you some insight into the non-specific binding of reagents in your system. And the next step is using these marker systems is to set this is your negative, so anything above here is your positive, and so using this you can analyse this population of cells here. Okay, so again, just coming back to the two colour dot plots, it's possible to measure the proportion of cells which are in these respective areas, and also how far along this axis they are, so as a measure of fluorescent intensity. And so the two key elements of a flow cytometry experiment when it comes to the data that you're pulling from it. Okay. So the two primary data outputs are the proportion of cells which are positive for a given antigen, and this is typically expressed as a percentage, but also the fluorescent intensity of that antigen. Now, in many instances, if you're just looking for the proportion of CD4 cells or CD8 cells in a mixed population, then this parameter would be fine. In some instances, it's also important to look at the fluorescent intensity, because you may be wanting to know, if we go back to our experiments when we were looking at CD69 expression, what we wanted to know was whether a particular molecule is having an influence on the capacity of cells to be activated. And so what we're interested in is how positive cells are for CD69. And so it's always worth remembering that the intensity data is a parameter which can provide valuable insight into functional responses. Now, a key issue that you should always consider is the potential presence of dead cells, because dead cells bind antibodies non-specifically, and this non-specific binding can masquerade as specific cell subsets. And the consequence of this is you can misinterpret your data. And so it's always valuable to include a viability or dead cell stain in your analysis. And I think if you don't include a viability stain, then I think one has to question the data that you're generating, because some of that positivity may well be due to dead cells within your particular experimental protocol. Now, there is a tendency to think, well, I need more colours, and so I don't have the capacity to include a viability or dead cell stain. The problem is, if you don't include one, then you don't really have an experiment, in my opinion. There are plenty of viability dead cell stains out there now which can be put onto particular laser filter combinations in your instrument, and so this is a key element of your experiment which you shouldn't forget. Okay, another piece of terminology which you may come across is spectral overlap. And actually this feeds into the concept of compensation. And my experience is that compensation, or the concept of compensation, remains one of the aspects of flow cytometry which continues to mystify new users. But essentially the concept is pretty simple. So what is spectral overlap? Well, spectral overlap occurs when the light emitted from one fluorochrome leaks into the channel which detects the fluorescent signal which is being emitted by another fluorochrome. So here we can see some of this emitted light being detected in this particular area here. So this would be provided as a sort of positive signal, which is not correct. The instruments these days, it is possible to eliminate this by electronically removing the signal, and this is the process termed compensation. But really you want to try and minimize this spectral overlap by the appropriate choice of fluorochromes in your particular experiment. And again, we will be coming back to this later on. The other element which is worth considering and keeping in mind is something called the stain index. And again, the concept for the stain index is quite straightforward, and essentially it is that some fluorochromes are brighter than others. So in its simplest terms, the stain index is a parameter which reflects the ability to resolve a dim positive signal from the background. So for example, if you're analyzing cells, it's better to choose a fluorochrome with a low stain index for parameters that are expressed at high levels. So again, keeping with the common theme of immune cells, antigens such as CD3 and CD4 are expressed at very high levels on the appropriate cell type. And so it's better to select a fluorochrome, a dimmer fluorochrome for those cells, and save the very bright fluorochromes for some of the surface antigens which are not expressed at high levels, or ones which are being induced by your experimental protocol. It's also important to be aware of the stain index when it comes to designing your antibody panel and minimizing spillover as part of your experiment. So I just want to highlight the concept of cell sorting. We have a number of very sort of high throughput cell sorters available to us these days, and essentially what cell sorting allows you to do is to identify cells which are passing through your flow cell either on the basis of forward and side scatter or on the basis of their expression of fluorescent molecules or a combination of both. And essentially what the instrument allows you to do is identify the cells in which you're interested. It will then put an electrostatic charge on the appropriate cells, and it knows exactly how long it takes for a cell to get to this point, and then the cells are put through basically through two electromagnets, and the appropriately charged cells can be directed into different tubes. It's also possible to separate cells into 96 well plates for cell cloning experiments or for subsequent functional analysis. And I think there's just one thing I would like to say before I pass over to Andy is just highlight that the acronym FACS is a trademark owned by Becton Dickinson and Company, and FACS essentially stands for fluorescent activated cell sorting. So certainly in the literature, if you're writing papers or presenting when you're not doing cell sorting, then it's more appropriate to use flow cytometry as a terminology rather than FACS. Okay, so I hope that's been helpful to you. It's very basic information, but it's important that you get the essentials correct. It's also important that you understand how a flow cytometry works, because there's a tendency these days for the new instruments to take away the need to understand, and it's actually becoming more and more important to get to grips with the technology that you're using so you can ensure that the data that you're generating and presenting stand up to scrutiny. Okay, so thanks for your attention. I'd like to pass the stage over to Andy Lane.
Great. Graham, thank you very much indeed. I'm sure everybody's found that really helpful, and that's a great foundation for the second half of the talk. And I'd like to focus more on the role of antibodies as tools and reagents, really to get the most out of modern-day flow cytometers. One of the points I'd start with is really in highlighting the power of these instruments. I've put a couple of references up here, and the first one, the paper from Stephen Perfetto, is actually now 8 years old, and this was a real benchmark paper at the time of its publication. It was talking about 17-color flow cytometry and using all of those different markers to unravel detailed subsets of the human immune system. Finding references more recently that are using multicolor flow cytometry for 9, 10, 11 or 12 colors is actually now relatively straightforward. And I've just chosen one particular one here from Altissier et al. Again, talking about dissecting human cell subsets using 12-color flow cytometry. A lot of the instruments, the modern instruments that Graham showed earlier in the talk, have the ability certainly to go up to 9 and 10 colors as benchtop machines. The slide I've put up here is really to highlight, however, one of the problems that comes up with the ability to undertake multicolor flow cytometry, and that is in relation to the availability of reagents, particularly directly conjugated antibodies. In this particular example, we're looking at a diagrammatic cell with 5 different antigens on the surface that people want to stain. They have antibodies available to each of those, but only 3 of them are available directly conjugated in commercial forms. 2 of them are only available in an unconjugated format, and that gives them a real problem in determining how they're going to undertake a multicolor experiment. A potential option which first comes to mind is the use of secondary antibodies, which will bind to these primary antibodies to give you extra colors and to detect these. But in a multicolor experiment, that becomes a real problem because these secondary antibodies immediately bind to the other primary antibodies, and in effect you simply have what is best described as a bit of a mess on the cell surface, and something that is very difficult to dissect when you're analyzing your flow cytometry data. What options do you have to overcome this? One of them really is to undertake conjugation of your antibodies yourself, or perhaps to use traditional antibody conjugation techniques by subbing this out to other companies. If you undertake this, there are a number of things that you need. Certainly, you will need experienced staff who have a certain knowledge of immunochemistry. Now, depending which dye you may be using, the knowledge that they will need of the chemical reactions to link antibody to a dye vary. To link fluorescein to an antibody is relatively straightforward. To link a phycobilioprotein, such as phycoerythrin, to an antibody is much more complex. You will also need a relatively large amount of your unconjugated antibody in purified format, typically perhaps 3 to 5 milligrams. Traditional conjugation techniques also invariably rely on having separation columns and potentially fraction collectors, etc. The separation columns are there to remove unconjugated antibody from conjugated antibody and also from free dye. And one of the problems that comes up from using those separation columns is that of yield. A typical yield from a conjugation experiment may be in the order of 70%. Given that you've started with perhaps 3 or 5 mg of antibody, you're losing significant amounts by these processes. These processes also take you a considerable amount of time. Again, the simpler techniques will take you a full day in the laboratory and plus another probably overnight period to finish the preparation. The more complicated techniques can take 2 or 3 days in the laboratory and a lot of hands-on time. Underlying all of this is the fact that the cost of undertaking traditional conjugation is very significant indeed. The cost comes from a number of areas. Firstly, you need the experienced staff. Secondly, you need relatively large amounts of your unconjugated antibody. And if you're buying that commercially, you may be paying perhaps several £100 or dollars per milligram for that antibody. And obviously you also have the cost of losing a good percentage of that on your separation columns. Even if you take some of the traditional conjugation kits that are available, these also include separation columns and you still come across the issues of yield. What I'd like to talk about now are the Innova Biosciences Lightning-Link kits, which are the world's easiest to use antibody labeling kits, which actually overcome all of those issues associated with traditional conjugation techniques. Lightning-Link provides you with a very simple one-step process, which actually only needs 30 seconds of hands-on time in the laboratory. The simplicity itself makes this technique extremely reproducible, and it's very easy to scale it from micrograms to milligrams. And being scalable from micrograms to milligrams underlines one of those other points, is that you actually can work with very small amounts of antibody and therefore reducing the input cost that you have. There are kits available working down to 10 micrograms of antibody. If you need to work on a larger scale, moving up to milligrams to tens of milligrams and higher is very straightforward to do. Also extremely important in relation to the Lightning-Link kits is the fact that you actually get 100% recovery of your antibody. These kits are optimized so that there is no need to use column separation technology. There is no reduction in yield from losses associated with that. The process is extremely simple. You take your label, which is in the kit from Innova Biosciences. You very simply modify the antibody with one of the reagents that is present simply by adding a small volume of that. Mix the antibody to the label. Incubate for probably 3 hours, overnight if you prefer. Stop the reaction and you have your conjugate ready. More recently, Innova have been able to further develop these kits to reduce the time from a 3-hour time scale actually down to something less than 20 minutes with a wide range of their kits. The process is exactly the same. You start with the Lightning-Link label, modify the antibody, incubate for 15 minutes, stop the reaction, and you have your antibody ready to use. You can literally have a conjugated antibody ready to use in less than 20 minutes in the laboratory. Graham showed this slide, or the table from this slide earlier on, really to underline the wide choice of flow cytometry dyes that are available nowadays. This is just a selection of flow cytometry dyes that are available in Lightning-Link kit format. And as Graham mentioned, these are grouped by the most commonly used excitation lasers. So there are dyes available to excite off of the 405 nanometer laser line, the very common 488 line, 561 lasers, or any of the lasers around about 500 nanometers, and also the 633 or 635 lasers. There are, within the Innova range, a number of other dyes which are suitable for use for people who want to use slightly different laser lines or want to design their experiments slightly differently. You'll notice that the Innova range includes some very traditional colors such as Fluorescent, which many people will be familiar with, phycoerythrin as well. Also, very importantly, these tandem dyes, which provide you with very, very large Stokes shifts and high wavelength peak emissions. And the wide range of dyes that are available means you have a wide choice for developing your multicolor flow cytometry experiments. Out of that list, how are you going to choose your dye? Now, Graham talked a little bit about this when he was talking about the structure of dyes and fluorescence earlier on. Obviously, one of the things you need to know is what laser or lasers do you have available. You need to choose a dye that will be excited by the lasers that are free for you to use. It's also useful to have an idea of the level of antigen expression. If possible, you want to use brighter dyes for weakly expressed antigens. Typically, a brighter dye would be phycoerythrin or allophycocyanin. You also need to know what other dyes are being used. Do they overlap with the dyes you want to use, or do you need perhaps to compensate your data or to change filters? And again, Graham talked about this aspect a little bit, and I'm just going to review some of that now. Using the same fluorescence overlap diagram, you can see in this case that the fluorescence emission spectra of FITC and RPE overlap quite considerably. You can reduce that using filters, and all standard flow cytometers are fitted with very good sets of filters, and the FL1 filter here looks at just the peak emission of fluorescein, the FL2 filter, the peak emission of phycoerythrin, and cuts out some of the overlap from fluorescein. However, there's still a significant amount of overlap, particularly of FITC emission into the phycoerythrin or the FL2 channel, and you need to undertake compensation to overcome that. This diagram shows an indication of uncompensated data and how this changes when you can correctly set the compensation settings on your machine. Several years ago, compensation would have had to be set during the acquisition phase of flow cytometry, which meant that you had to get the settings absolutely right. Much more recently, most of the software available for analysis actually now allows you to undertake compensation within the software after you've acquired your data, which is a significant advantage over a few years ago. Anyway, this is typically uncompensated data. You can see that FITC stained cells along this axis are overlapping into the phycoerythrin channel on the y-axis. So these cells are not actually expressing any PE labeled marker. This is literally overlap. And using the electronic settings on the machine, you can begin to remove that overlap in stages. And you'll see this is an example where compensation has been applied but perhaps not enough, and this is data that is undercompensated. And if you move through to here, this is correctly compensated data. So you can see that this population is positive along the x-axis, but not positive on the y-axis. And similarly here, y-axis positive, x-axis negative. It is possible to overdo this. You can overcompensate your data and actually move this out of the threshold of detection, so you want to be aware of that as well. Filters are also important, but I think it's still, even with modern machines, very true to say that filters within flow cytometers are rarely changed by the user. It's possible in some instruments, and it might be useful to get the best performance from a particular dye. In most cases, you'd want to take advice from the manufacturer of the flow cytometer, both about the actual technique of changing the filter and also of the best filter for the dye that you're using. I just thought I'd mention a couple of bits of common nomenclature, one of which is the so-called bandpass filter, which in this example, a 530/30 BP filter is a filter which detects light in the range of 515 to 545 nanometers, so plus or minus 15 nanometers. There are also long pass filters which only let light through that is longer than a certain wavelength. So a 650 LP filter will only let light above 650 nanometers through. So typically, if you had a 650 LP filter in place, but you wanted to detect a dye that had a peak emission at 640, you would need to find an alternative filter. And as I said, your manufacturers are often able to help you in some of these cases. They will be very familiar with the dyes that are commonly used and are able to offer you those alternatives very straightforwardly. Changing tack just a little bit, I talked about the fact that the Innova Lightning-Link kits are very, very simple to use. What I'd like to take just a moment or two to consider are some things that you need to think about when conjugating your antibody and about the antibody itself. Commercially available antibodies actually come in many forms. and you may need to check with your supplier about some important details. Some of these are outlined here. Firstly, ensure that your antibody is pure. Make sure that other proteins have been removed. So for instance, a lot of antibodies are now grown in tissue culture supernatant and are purified with a protein A or protein G chromatography step. However, some are actually sold in the format of the tissue culture supernatant, and some polyclonal antibodies may be sold as unpurified serum. So make sure you're clear that purification has taken place. And my comment here is also relevant. Make sure that the proteins that have been removed haven't been put back again afterwards, which may seem to be a strange comment. However, a lot of antibody manufacturers, especially if the antibodies are being sold in a relatively dilute format, will put a protein stabilizer, most commonly bovine serum albumin or BSA, back into their purified antibody preparations to undertake that stabilization. And that may well interfere with your conjugation reaction. So look very carefully at your product data sheets to make sure what other stabilizers may be present. It's also important for the kits, for their best performance, to have antibodies at a concentration of around 1 milligram per mil. Again, check on your commercial antibody data sheets to find out what the antibody concentration is. And finally, be aware of what your buffer formulation is. Most common formulations of commercial antibodies will be suitable for use. Most of them are presented in a phosphate buffered saline. But do be certain that amines such as glycine are truly absent, and make sure that phthiols such as DTT and mercaptoethanol are absent as well. It's worth pointing out with glycine that if you're undertaking a purification on protein A or protein G, you may well be using a glycine buffer to elute the antibody from the column. The next step should be to fully remove that glycine with an extensive dialysis step. If that dialysis step hasn't been undertaken, or perhaps hasn't been as extensive as it may be, there may still be some glycine present which may inhibit the reaction with the Lightninglink products. Tris is actually okay in your buffers up to the level of around about 20 millimolars. So if you have Tris in your buffers at low level, that's probably not going to be an issue. Quite often, commercial antibodies might not meet those criteria. And Innova are able to provide you with a purification kit to purify, concentrate, or simply change the buffer of your antibody. And on our website is this flow chart which will help you decide if you need to undertake further purification. So you can check if your antibody is purified, if the buffer contains something like BSA, if it contains amines such as glycine, and whether it meets concentration requirements. If all of these are met, then you're absolutely fine to carry on and use your antibody straight away. Otherwise, there are a number of slightly different purification kits that are available for you to use to undertake purification and cleanup of your antibodies. These will all work on small amount of antibodies and are very straightforward and simple to use straight before undertaking the conjugation with the Lightninglink kit. Once you've undertaken the conjugation, what are you going to do to use your new conjugate? The thing I'd like to underline is you actually will use them in exactly the same way as your normal staining technique. However, you may need to titrate your antibody, and possibly extensively. Again, it's worth pointing out that many commercially available directly conjugated antibodies are probably sold around about 10 to 100 micrograms per milliliter. You have just conjugated your antibody probably at 1 mg per mil, and as we said earlier, there is no loss of yield, so your antibody is still going to be relatively concentrated. Therefore, you want to undertake a series of dilutions to find the best performance of your antibody after conjugation. It's not unusual in my experience to find that you need to take that down to 1 in 100 or 1 in 200 dilutions. This very much depends on the antibody and the antigen you're using, so there's a lot of variability here, but be prepared to undertake that experiment to get the best performance from your new conjugate. Storage of your conjugate. The most reliable is to store at 4 degrees in a concentrated format. It may also be useful to add a preservative such as sodium azide, and if you wish to store your antibody diluted, a carrier protein such as BSA, as I mentioned earlier, would be useful. Be aware that you can safely freeze some conjugates, but others you shouldn't freeze at all, and never freeze phycoerythrin, allophycocyanin, or any of the tandem formats of dyes. If in doubt, you're better off to not freeze a conjugate, but to keep it at 4 degrees. And finally, keep your conjugates away from light, particularly tandem dyes. They're especially sensitive to light. And that will hopefully, by the time you've made your conjugate and you've followed these rules for using them, you'll be able to undertake new multicolor flow cytometry experiments with colors and conjugates of antibodies that aren't easily available, and make the most of the flow cytometry instrumentation that's available. I'm just going to pass over to Claudina very briefly now. We're going to try to answer some of your questions, and I think also Claudina just wanted to mention something else about a show that we will be at in the next few days. Thanks very much for your attention, everyone.
Well, thank you very much, Graham and Andy. It is now time for our Q&A session. I just wanted to quickly remind everyone and those who might have missed the first minute or so of the webinar that if you do have any questions, please do type them in. The question box is in your control panel, and if you can't fully see it, you can press the little plus button next to it and it should open it up. So any burning questions, please send them in. I'd just really briefly like to tell you that we do welcome any feedback you have for us, and we'd very much appreciate if you let us know how we can improve in the next episode. If you have any suggestions for future webinars, please send them in, and we will see if we can tailor them to your interests, as we do want this series to be a useful educational resource. In addition, if you would like to discuss this further, Helen, my colleague from Innova, and I are going to the AstraZeneca LRIG Pharmaceutical Flow Cytometry and Imaging Conference on October 10th. There will be plenty of flow-related talks, as well as ones on biomarkers, personalized healthcare, and the others that you can see on this banner. If you're interested, it's free for delegates to attend, and also we'll be exhibiting there, so if you decide to go, please stop by our booth and say hello. Please bear in mind that this webinar is recorded, so if you missed anything, don't worry, because we will upload it to our YouTube channel in the next couple of days, so you can always come back to it. You can find our YouTube channel at youtube.com/Innova Biosciences. If you would like a PDF copy of this presentation, also please get in touch. I'll be able to send it over. You might also be interested in our short flow cytometry guide, which I can also provide in PDF form by email or send over a hard copy in the post. In order to contact us, my email is provided in all the registration and reminder emails, or alternatively, you can email us at info@innovabiosciences.com and we will get back to you. And that's enough from me, and let's move on to the Q&A session.
Okay, so we have quite a few questions actually, so we'll try and get through as many as we can. I've just noticed one here that you might be able to answer, Andy.
Okay, Graham, you had one in mind, I suspect. Is that the most recent one that's come through?
No, this one is from Nina, and Nina's asking really whether the Lightning-Link kits can be used to conjugate antibodies for in vivo use.
Okay, that's an interesting question, actually, Nina. There's no reason, there's nothing within the Lightning-Link kit that would cause you a problem in itself for in vivo use. It's not something that we actually have this tested for. Basically, the Lightning-Link kit makes your conjugate in exactly the same way. You have an antibody. You're not adding any preservatives. There's nothing toxic in there, but that's not something that we've actually tested it for. I see no reason why it would cause you any problems.
I think what's also worth mentioning even is that we in our lab have used the kits to label proteins as well, and we can use Lightning-Link label proteins in confocal microscopy, so the proteins can be internalized as well appropriately.
Okay, that's great. Thanks for that, Graham.
Okay, so there's also been a question. I mentioned during my section of the talk about being able to measure fluorescent intensity, and that gives you some indication as to how bright an antigen is expressed. And we've been asked whether it's possible to quantitate that brightness and so put an actual figure on it. And yes, this is possible to do. There are a number of commercially available beads which have a defined number of binding sites on them, and so you can calibrate your instrument and actually work out how many antigens are on the surface of your target cell of interest. So yes, this is possible. Okay, we also, yep, go ahead, Andy.
Okay, thanks, Graham. There was a question from Erin regarding the Lightning-Link times moving from 3 hours to 20 minutes, or the kits that I mentioned in relation to that. The standard Lightning-Link kits have that 3-hour incubation time. You will see, if you go to the Innova Biosciences website, you will see a series of so-called rapid Lightning-Link kits, which are a new generation of kits available to undertake the 20-minute conjugations. So the format of the kits is really exactly the same. They've just been formulated to allow you to reduce that incubation time to as low as 20 minutes.
Okay, and we have a question from Ashim all the way in Manitoba in Canada. He's wanting to know how to overcome autofluorescence. In actual fact, Ashim, I mean, autofluorescence is an inherent state of the cell, so it's quite difficult. In fact, cells such as dendritic cells, for example, have their own sort of typical autofluorescence profile, and so you can actually start to look at using autofluorescence as an indicator of cell type. What you have more control over is trying to reduce non-specific binding, and that can be done by either blocking the FC receptors, which could allow this FC receptor binding using monoclonal antibodies, or blocking with irrelevant immunoglobulins, or indeed just having sort of protein in your staining solution, BSA or fetal calf. That will reduce the non-specific binding of your antibody. But it does highlight the need to have your appropriate controls in so that you're aware of what level of non-specific binding you have in your system.
Okay, thanks, Graham. There was also a question from Diana asking if we had a list available indicating the relative brightness of the different dyes. And Diana, that's something that actually we can send you by email over the next couple of days to give you some indications of exactly that information. And another question from, I think it's Shawan, wanted to know if you need to purify and separate unconjugated antibody or dye within with using the Lightning-Link kits. And probably one of the main advantages of the Lightning-Link kits is that you actually avoid this column separation process. The kits are set up in such a way that there's no need to purify and separate out unconjugated antibody. Your resulting product after you've used the kit is conjugated antibody, and there's no other side reactions that you need to be concerned about.
Okay, thanks, Andy. We have a question relating to whether or how flow cytometry can be used for measuring apoptosis. Well, there are a number of ways for measuring apoptosis, and actually if you are interested in apoptosis, what you really need to do is do a number of different techniques. And so I suppose the classic examples would be to use flow cytometry, assessing flow cytometry binding of annexin 5 to assess the redistribution of phosphatidylserine onto the cell surface, which is associated with apoptosis. One can also use antibodies which recognize the active form of caspase 3, particularly within the cell, but it's always best to use a number of techniques. In actual fact, what is attracting a lot of interest now is actually the role of autophagy in a lot of biological processes, and there are now techniques now for looking at autophagy using flow cytometry. So I think there are lots of options available for you.
That's great. Thanks, Graham. There was one other question here from Tom about the benefits of direct antibody labeling versus secondary antibody labeling. And this is quite an interesting area. Actually, there's probably over many years been a perceived wisdom that using a secondary antibody labeling system, either in flow cytometry or indeed in other techniques such as Western blotting, etc., will lead you to increased sensitivity because you're using 2 layers of antibodies. We've got quite a lot of data that actually shows that using direct conjugates can actually be just as sensitive because you're not having to worry about non-specific binding with your secondary antibodies or cross-reactions of your secondary antibodies with the cell surface or any of the other antigens that you're using. It's actually, the main advantage is that direct labeling is indeed a much cleaner technique, and you can get results at least as good with direct labeling as you can with secondary reagents. Again, on the Innova website, there are some examples doing a comparison of direct and indirect antibody labeling, which show just that. Okay. Well, I realize, everyone, we're coming up towards the end of the time for the webinar. There's a number of other questions that we haven't had time to get through. A number of people have asked for copies of the webinar, etc. The webinar will be available on our website very soon. We will get back to any of your other questions by email within the next couple of days. And I'd like to thank all of you for attending and taking your time out to listen to Graham, myself and Claudina today. On behalf of Innova Bioscience, I'd like to thank Graham for joining us from Chromocyte and for your help and explanation of flow cytometry at the beginning of this talk, which I'm sure everyone has found very useful. So thanks everyone.